We are joined today by Prisma Health's Dr. Alan Litwin, Vice Chair of Academics and Research and Professor of Medicine at Prisma Health, University of South Carolina School of Medicine, Greenville, and the Clemson University School of Health Research. During the COVID pandemic, Dr. Litwin has led the Prisma Health Rapid Innovation Task Force, and that's what we're going to talk about today. So we've got Dr. Litwin here to moderate a discussion, including Cody Reynolds, the Technology Transfer Manager of Prisma Health System. We've got Jerry Chang. You can wave there out there, Jerry, who's the Managing Partner and COO of Samaritan Biologic, as well as a number of other Innovation Task Force collaborators. So, Dr. Litwin, thank you for your leadership in this. I'll hand the mic over to you.
Great. Well, thanks, Michael, for this opportunity to highlight some of our innovations. The COVID-19 Rapid Innovation Task Force is a multidisciplinary team that includes leaders from Prisma Health, the Health Sciences Center, and that includes Clemson, USC, and Furman, and industry. And it was designed and is designed to discuss real-time problems identified from clinical and operational leaders in the field, to develop solutions collaboratively, and to link to academic and industry partners so we can rapidly operationalize. We convene project teams working in key areas, including novel therapeutics, public health, PPE or personal protective equipment and sterilization, ventilation and airway, novel ideas, testing, as well as business collaborations. And during the pandemic, our group has worked closely with the Prisma Health Incident Command Team. Many of us are members, as well as our executive leadership to rapidly take control of the pandemic. As a brief kind of introduction, most healthcare organizations have been largely dependent on GPOs or group purchasing organizations. And during pro-COVID times, these GPOs had no problems handling our day-to-day operations within healthcare. But with COVID-19, it quickly became apparent that we can't solely rely on these one-stop shops for critical supply needs, and the usual innovation timelines needed to be sped up. So again, the Rapid Innovation Task Force allowed us to become more nimble and quickly identify critical needs, work with our local innovators in industry, and develop and implement solutions for South Carolina to protect and save the lives of our frontline workers and all of our citizens. So it's been an honor to work with many innovators in our ecosystem, and today we're going to hear from 4 groups and 7 innovators, and each group will answer 4 simple questions. These questions are, what was the problem? What was the solution? What inspired you? What was your personal motivation to solve this problem? And how did the collaborations help with the process? And again, I apologize in advance to each group if I interrupt, because we only have 10 minutes for each, so that each group has enough time and that we allow a few minutes for questions at the end. So we're going to jump right to it. Looking forward to the presentations and discussion. We're going to start with Jerry Chang and Cody Reynolds, who will discuss business collaborations.
So, hello, everyone. By quick way of introduction, I'm Cody Reynolds. I'm the Technology Transfer Manager for Prisma Health. And in that role, I'm also responsible for reaching out to local businesses and also our academic partners to figure out what we can, what talent we can leverage to try and solve the problems that we're facing. And I think that Jerry Chang has certainly been a more than exemplary example of a great partner in industry that's helped us solve a lot of problems, of which just a few we'll address today. So, Jerry, if you wouldn't mind introducing yourself.
Hi, my name's Jerry Chang. Again, as Alan mentioned, I'm the COO and Managing Partner of Samaritan Biologics. I've been an entrepreneur pretty much all my life, and I've been innovating and developing different types of products for the healthcare industry over the past 20 years. So it's been a great privilege to work alongside Prisma and, again, all the different academic institutions and local businesses as well towards hopefully solving some of these issues that we're seeing with COVID-19.
So really quickly, just the standard format that we're going to try and adhere to for all the subcategories that we're presenting today is: what was the motivation or the inspiration for solving a problem? What were the problems that we faced? What is the solution, or where is that solution stage at? And then how did the partnerships with Prisma Health, our academic partners in the industry, facilitate the solution? So My motivation personally is fairly obvious. I work for Prisma Health, and I'm trying to ensure that it has all of the PPE, the testing, and the insights that it needs to serve its community and to reach out to everyone else. So that's my day job. But I believe that Jerry Chang went above and beyond to really help his community. So, Jerry, what was your motivation?
Well, in my particular case, my motivation was a little bit personal, as my wife is a physician as well as an employee of Prisma Health, and the fact that all of my friends locally and around the country are physicians or in the healthcare profession, I think that meant a lot to me. And again, as an entrepreneur and having started multiple businesses, we always try to develop, provide, or create some type of product or service that helps improve the lives of the community or other people. And when COVID hit, given that I've had experience designing and developing products, I thought that as long as I had the means to either source the delivery of PPE or other needed supplies, or had the ability to help in the design and delivery of these types of products to keep our healthcare workers safe and our community safe, why wouldn't I do this? I think that's just the right thing to do. That was my personal motivation.
So moving on, we had several problems that were identified nationally as potential shortages back in March and April when the Rapid Innovation Task Force first started. And one of them was face shields and eyeglasses. So, Jerry, these are your photos. This is what you experienced in sourcing materials for these. Can you give us a little insight on that?
Oh, sure. I mean, just, during the COVID pandemic, I think everybody experienced this. These are pictures from local warehouses and grocery stores, and you can see there were shortages of food as well as critical supplies. You all heard about the shortage of appropriate N95 masks, the ability to have appropriate test kits, respirators, ventilators, the great toilet paper shortage of 2020 as well. It was just difficult. So we had to go outside the normal supply chain to try and source all these critical needs. And it was great, again, to work with innovators across the state of South Carolina and our community to help solve these issues.
So, Gary, again, these are your babies here. There's a lot of different face shields here that you went through. Can you talk about that?
Yes. So just to give you an example of how this worked, as Alan mentioned, or Dr. Liquid mentioned, the hospital system identified a critical need and a potential shortage. And even though they've been phenomenally prepared, as they've been preparing for COVID hitting, before even January of 2020, we wanted to be ahead of the game, and we knew that this would be one critical piece of equipment that would be necessary for our healthcare workers. And so we partnered with, again, Furman, Clemson, USC, as well as the Prisma clinicians to identify the concern, the needs, and what type of specifications we needed. And through their help, we were able to prototype a variety of different options that the clinicians were then able to go in and evaluate either within their test labs or, based upon their experience, they put us to identify a specific design that they would like to have. And if you go to the next slide, so the next phase would be, the prototyping and testing. So this is where we reached out to local businesses because, although we had an idea of what we needed to create, there were issues getting these needed supplies because, as you can imagine, worldwide, there was a shortage of basic materials, just as like elastic for straps for your face shields, the films for the coverings. So we worked with, again, local suppliers to identify, what options existed. And we created a number of different prototypes and, again, had those prototypes evaluated by the clinical staff at Prisma. And once they chose, their favorite option that met all their clinical needs. If you go to the next slide, Cody. Oops. We went ahead and developed the process in which to manufacture these. And so you can see here, we worked with a lot of different folks, again, local universities, the supply chain team at Prisma, American Foam and Fabric. Their normal business is supplying foam and fabric for the automotive industry. Relco did the die cutting. Mitsubishi polyester film was able to supply us the anti-fog film necessary for these shields. Paper Cutters was able to cut them for us, and we actually had the South Carolina Department of Corrections perform the assembly. So all in all, it was a team effort, and we were able to produce the materials necessary for over 375,000 face shields at less than $1 a piece. We're actually currently doing the same thing. We followed that same process, safety glasses with disposable eyepieces as well. We're working with some of the partners that we tapped into in the prior project, and we're on track to deliver over 100,000 iframes and 300,000 of the disposable eye shield pieces for less than 50 cents a unit. And lastly, this is our most current project. It's not put in production yet. This is a prototype of a new semi-reusable face shield. This particular model is 3D printed, but we're modifying that design so that we can injection mold it and produce it in mass much cheaper.
So I think everyone can read the bullet points here, but I wanted to take a second and just really acknowledge Jerry Chang's company, Samaritan Biologics, and particularly MSI Mold Builders. Jerry might want to throw in a few more names too, for having either donated entirely all of their work and materials or offered them at cost just because of the size of the need that we had. So again, thank you very much, Jerry, MSI Mold Builders, Samaritan Biologics. Jerry, any others you want to call out in particular?
Oh, Paper Cutters, Mitsubishi, they're really refreshing, working on this project and working with this team. It was a great reminder that there's still a lot of people out there trying to make the world a better place. So it was, again, it's been exciting, a lot of fun, and just glad I could be part of it.
I couldn't say that I agree more. It was great too, just that all the normal barriers that you might expect to talking with between industries and vendors and buyers, all of those barriers came down in order to make the product available. So thank you very much.
Great. Well, thank you so much, Jerry and Cody. We're now going to introduce Drs. Wesley Liao and Chris Cole, who are going to discuss innovations in PPE. And we're a little bit ahead time, so I think you needed a little more than 10 minutes, so feel free to take up to 15 minutes if you need it. All yours, Wes.
Awesome. Good afternoon. I'll just wait for those slides to come up here.
Yeah, Cody's getting them on right now.
And of course, it's just a little bit more complicated. There we go.
Thank you so much. So for our brief presentation today, Chris and I just wanted to focus the discussion around PPE and specifically the N95 masks. So we'll go to the next slide here. And so just by way of, I guess, a little storytelling, I am Dr. Liao, anesthesiologist by trade, vice chair with our department here, and have been very engaged in the Rapid Innovation Task Force since the inception. It's been an incredible experience. One of the issues that our perioperative services and then, just more broadly, the entire hospital system has been dealing with has been how do we make sure that we have availability of really high performance N95 type masks in a pandemic situation. So we are dealing with issues with worldwide availability and more importantly that the demand is certainly mismatched relative to worldwide supply. And, not all N95 masks are created equal. And so what we wanted to really make sure was that we were providing high quality, and consistent products to our frontline team members to make sure we were protecting every individual, and at the same time making sure that they're able to not only be protected from the patient, but also protecting families when they go home. So because of, oops, let's go back to the previous slide there for a second. Great. So because of the issues with sustainability of these masks, we really needed to look at alternative options. And knowing that the mask shortage globally could be a rate limiting factor in terms of just how operationally successful we would be as a health company, we had to look at alternative means. Really three main things: looking at reprocessing or recycling of the existing inventory, and then looking at alternative mask products. So thinking outside of the box of what a normal N95 mask design would be. And then additionally looking at new technologies that would be again something that we could augment our pre-existing N95 supply to make sure we have sustainability of this precious resource. Furthermore, what we realized too early on in this is that a lot of the alternative mask products available on the market internationally, such as the KN95 product, while initially we thought we could get our hands on a lot of it and the N95s or KN95s wouldn't be a problem and we'd have the mask situation covered, we found very early on that through our testing and our collaboration with Dr. Cole and Clemson, that this actually was not going to be a suitable replacement or alternative for N95 masks. And it was an amazing and I think a very critical catch and decision that we made through very, very scientific and quantitative testing measures. We were able to define that that product was not safe. It was probably good for civilian use, but certainly not for the healthcare setting. And it's remarkable. I was just talking to Dr. Cole last night, but it's remarkable that now our literature has sort of caught up with some of the observations months later, things that we had found out and discussed in April and May are just being described now. Next slide, please. So the task force really, when you look at masks, looked at three main issues. When we looked at reprocessing, and I'm going to talk about our processing pathway in just another slide or two, we looked at reprocessing issues. What are some of the impacts of the reprocessing operations, specifically as they pertain to impact on the mask material, and how can we really understand and quantify the impact of that reprocessing cycle? The other thing we looked at was just performance verification. So how do we know that these masks are performing the way we want them to after 1 cycle, 5 cycles, 10 cycles, or even more? And understanding that there have been some tests with different technologies to understand the limitations and impacts on the materials portion. And then finally, when we look at the alternative mask designs and we look at other technologies to augment our inventory, how can we really identify the mask and filter media performance characteristics? And really, how are we going to be able to measure these to make sure that they're going to meet the standards and provide the critical levels of safety that are absolutely required for our frontline team members? And particularly in the setting of a pandemic, how do we get access to test these things in a very time-sensitive fashion so we can make quick decisions and continue on with working with viable designs and be able to separate viable from non-viable designs? Next slide, please. So after extensive research, discussion, and consideration of all of the different pathways for reprocessing and sterilization of masks, which involve looking at basic things like putting them through a washing machine and physical tumbling of the masks, to looking at exposure to UV, to hydrogen peroxide vaporization. As a healthcare organization, we settled on the hydrogen peroxide vaporization decontamination. And for several reasons, we thought that this was going to be the best fit for us. Number one, it's been an industry standard process, so it's already well established with research institutions in the pharmaceutical industry and in many large academic medical centers, including Yale and Duke, who have published on their utilization of this technology. When you look at hydrogen peroxide as a material itself, this hydrogen peroxide is very, very, very low in its toxicity. And after it's exposed to masks, it really breaks down into simply water and oxygen. So we know that, you know, this is a very favorable thing when you compare it to other potentially more volatile agents. It's relatively easy to use with the machine that we have. It's very elegantly able to vaporize the hydrogen peroxide in a room that is of proper size. And we've had, you know, direction already through some of the academic institutions. As I pointed out before, Duke and Yale have described this in the literature. That's been out there for over a decade, so we really felt good about this. To date, since we've been doing the reprocessing project between our two markets in Greenville and in the Midlands, we're proud to say that we have processed over 125,000 N95 masks, and now they sit in our climate-controlled warehouse where they're ready, and we hope that we'll never need to use them, but if we do, we have a great option to deploy them out to the front lines. Next slide. And finally, just a little bit more detail on our process here. So basically, we collect our N95s through a collection bin at many clinical areas. And once we get it, transport it to a centralized processing location, there are many points where we are doing physical inspection and sorting of these masks. And we're looking for soiling issues and physical defects, and even things like, you know, strap integrity, making sure there's no makeup on these masks. So there are multiple points of inspection. Once we deem that these masks are good enough to actually be reprocessed, we're able to put them on a hanging rack, and the hanging rack goes into a very specially prepped room. In that room, our hydrogen peroxide vaporization machine is able to be turned on, and once that vapor gets to a certain level, we're able to allow it to basically penetrate into the mask fabric. And we are able to very consistently determine that we've had a reliable cycling and penetration of the hydrogen peroxide through both biological and chemical indicators for every single cycle that we put through. And this gives us assurance that we have done a great job and that we have high quality control. Finally, the masks are let to just off gas so that there is no unusual or offensive residual odor from the processing. And once we are done with that, they are again scrutinized to make sure we've got good structural integrity and to make sure nothing in that processing has adversely affected the mask. And once we've gone through all of those visual inspections, they are then put away and then transported back to the MDC for safekeeping. And we do pull masks off the line after they're done, and we do examine them again for all of the positive characteristics that we're looking for, including sending them off to Dr. Cole's lab to make sure that they're meeting our expectations. Next slide. I'm going to turn it over to Dr. Cole to kind of talk about our testing and approvals.
If you think about the tests that Wes has just mentioned, the biological and chemical indicators that are run with every load, and then the post-biological testing on the Petri dishes to see if anything grows off of random mass, you appreciate that those tests are really checking to make sure that the masks are clean, that they're sterile. But there are none of those tests that tell you that the mask actually will function to protect a healthcare employee. And that's where I came in, was trying to figure out what kind of tests we might be able to use to verify the actual performance on the N95 masks. Well, there are a whole series of tests which are used right now in the development of an N95 mask design, as well as for quality control and manufacturing. The problem is many of those tests are designed to be used on the filter media, that two-dimensional white fabric, before it's converted into a three-dimensional mask. So a mask doesn't lend itself to go in those kind of test rigs. But in addition, a lot of the test rigs aren't designed to be used with masks that might have gone through a recycle process. The other problem is that although there are really good labs that developed many of these test methods and do a lot of the testing, we found that you had months-long backlogs trying to get anything through them. So the bottom line was nothing that appeared to already be in the market was set up to be able to be rapidly used for testing lot by lot the performance of the masks that were coming through the vapor hydrogen peroxide process. So instead, what we looked at, we could see the next slide, we looked at what are some alternative things. And what we finally latched onto was the fact that in the United States, everyone who wears an N95 mask must undergo an annual fit test. So what the fit test is doing is it's making sure that that mask style actually properly fits the wearer. And in that testing, it's actually testing the filter media that makes up the mask design, and all the mask components, such as how well does the nose wire work, how well do the elastic straps work. So everything from the wearer back through the mask itself is being tested in a fit test. Well, it turns out that Clemson University actually does a lot of this kind of mask test work continuously. You may be aware that Clemson University actually runs the emergency services for the city of Clemson as well as for the university. So we constantly, pre-pandemic, were testing people and fitting them with N95 masks of different designs. We do testing on what's called a quantitative fit test. There are a series of 4 activities which are designed to stress the attachment of the mask to the wearer's face. You're collecting a signal which basically tells you how much of a sodium chloride aerosol is escaping through the mask, either through the filter media or through a seal failure to the face. So you get quantitative numbers. You got to score at least 100 to be able to pass. But it gives us a numerical feedback for every one of the tests, as well as an overall feedback. And the challenge, the sodium chloride aerosol particle challenge, uses 40 to 70 nanometer diameter sodium chloride particles. Those particles are actually slightly smaller than the 90 to 120 nanometer particle size for the coronavirus itself. So you're challenging with particles that are actually slightly smaller than the actual virus particles, not just the virus particles as they might ride on a water droplet. If we could go to the next slide. So what we find using our test protocol is we've never tested any of the 3M 1860 mask. That's that aqua blue mask that you frequently see healthcare workers wearing. We've never found one of them, whether they're new or whether they're after biocoil processing, failing that minimum 100 fit factor. The flip side is we've never found a KN95 that'll pass a fit test. And that's even after the KN95s we got our hands on, after CDC went through and culled down the list of manufacturers that they said gave them masks that it at least would give them particle filtration. So our feeling is that quantitative fit testing is extremely useful for looking at the biocoil reprocessing, but it's also extremely useful as a development tool. So part of what we've also done is go in and look at new filter media, we've looked at new mask designs, and we've needed to be able to test how good those designs were. Did they fit our test subject? We find that quantitative fit testing is absolutely the way to go. In about 5 minutes, you get whether that mask is going to work at all. We could go to the next slide. We've looked at a lot of other designs. These tend to be a combination of filter media with hard elements, so they're more like the P100 or the N100 kinds of masks, the PAPRs and so forth, working with individual entrepreneurs around here. Along the way, we've actually found that there are 2 South Carolina companies which manufacture today an N95 filter media, which isn't in the market. So we've got a lot of potential to be able to grow the manufacturing industry in South Carolina. We also build a lot of different kinds of masks. The mask that you see on the slide, which is the bottom left, this is the mask we make. We've made hundreds of these things for the Solid Orange people at Clemson's ICAR unit over in Greenville. We've made these for a lot of different people. It's just a matter of picking the right fabric. In this case, it's one of Milliken's fabrics designed to give us the right properties for a cloth mask. Some of the members of our team have pointed out that personally, their biggest problem was finding masks that would fit their young children that have small size faces. Those aren't readily available in good masks on the market. So the bottom right-hand side has a couple of those masks that we've designed, and they're sampling it with their friends. One of the masks, disposable masks we've got on the upper left, this is actually just a single layer of the N95 type material, just made as a disposable mask. It's acoustically, it gives us the best performance that we've ever gotten on a mask. We've got some other designs that we're not showing right now because we think that they're potentially patentable. It's been a matter. People come, they recognize that there's a capability here. There's an absolutely wonderful partnership between Clemson and Prisma Health, where they've got the interesting problems, and hopefully we're helping them with some of the solutions.
Great. Well, thank you. That was fantastic. Appreciate that, Chris and Wes. Let's move right into Drs. Phil Michella and John Dardeen. No slides, so it'll be a free-flowing discussion. If you would introduce yourselves first, all yours.
Sure. I'm Dr. Michella. I'm one of the emergency medicine physicians that works at Prisma Health. I'm also one of the assistant research directors for the Department of Emergency Medicine, and also sit with Alan as part of the Rapid Innovation Task Force, and also a part of a subcommittee called the Novel Ideas Committee. We kind of set up a group of nurses and physicians and pharmacists and everything like that to try to vet the system for various ideas coming from anywhere and everywhere. Basically, we put out solicitations and try to get ideas that come in and try to vet those across a multidisciplinary team to then pass up to Rapid Innovation Task Force. One of those ideas came from within the Department of Emergency Medicine with myself and Dr. Ben Roth and Dr. Chris Joffrey, where we kind of in our garages tried to come up with a portable negative pressure environment that I hesitate to show you the pictures because they're so crude and funny. But through the collaboration with Clemson, we got connected up with Dr. Desjardins in the Department of Bioengineering, and with some of his senior students as part of their classwork to try to bring engineers and physicians together to try to tackle this sort of complex problem. The best thing about this is that, you know, we're clinicians, so we understand what we need, and then the engineers actually understand how to do that. And so a lot of times it's hard for the engineers to come up with an idea on their own because they don't really know what are the physical specifications that are necessary for a medical device. And so through our unique collaboration, we actually brought the engineers and the students over to our simulation center and actually performed several of the procedures, talked through the sizing, taking measurements during the different procedures to make sure that whatever device we were going to make was going to actually be functional. And then worked through some of the specific parameters on the sizing for whatever device would be necessary to sort of generate the negative pressure. So with that, I'm going to turn it over to John, and we may go back and forth, but we hesitated to show you pictures, A, because the original ones would make you laugh, and then B, the other ones are in current patent applications, and so we didn't want to divulge anything that might jeopardize that.
Thanks, Phil. I'm John Desjardins. I'm a professor in the bioengineering department, and my day job is helping to direct our senior design program in bioengineering. And fundamentally, that's all about finding unmet clinical needs and working to develop prototypes and test those prototypes out towards the development of a finalized biomedical device. And Prisma has certainly been an active and gracious collaborator with us for the last dozen years in that respect. And we have had many, many collaborations between our students and faculty and the clinicians at Prisma and across the state, honestly, in the design of medical devices. And so when Phil came to us with a problem prompt, it was really quite a seamless integration of our students and our skill sets in that regard to start whittling down possible solution sets for this. And quite fundamentally, the inspiration, the need came from trying to protect healthcare workers and the environment that these positive or potentially positive COVID patients were inhabiting, and keeping the virus as contained as possible while still being able to administer clinical care and not contaminating the entire hospital in the process. So we really just took the idea of you could either think of it as a larger mask around the patient that had a negative pressure for filtration, or a smaller environmental room or a positive pressure or negative pressure room, and isolating that COVID environment to just around the patient while still being able to do clinical procedures. So for us, it was a pretty straightforward process of doing some prototyping. All the while, this happened over the summer, and so there was very little availability of, as others have said, materials, parts, and supplies. So this entire thing was sourced within, Target, Walmart, and Lowe's. We became frequent flyers with them, and went through easily a dozen, if not 2 dozen prototypes. And as Philip said, iteration is key in this regard. We don't know what we don't know, and so we had many frequent visits to the simulation center and to our clinical partners to troubleshoot solutions. Ultimately, this thing is essentially a fume hood that you put a patient in, or at least the patient's head. And so we operated on that principle of a negative pressure environment with airflow to limit potential exposure of the patient to the environment. And came up with quite an elegant solution, I guess. And it's been on trial there at Prisma. And ultimately, it basically just sucks air in from around the patient through HEPA filters and back out into the room without need of intensive infrastructure modification or potentially limiting the mobility of the patient or limiting the clinical procedures that could be done on the patient while in this small covering area.
I think what's very important to note about this collaboration back and forth as well is that we've taken what was already sort of published a little bit in the literature. So when this pandemic first came out, there was a bunch of what we call passive devices, which kind of hit the market and hit the research literature and everywhere, which are basically sort of rigid boxes or rigid drapes, which are nothing more than a glorified bigger, face shield around the patient. And in subsequent testing, when physicians were otherwise doing procedures and things like that, through simulated environmental testing of these, it actually shows that may actually increase the rate of infection. And so from the beginning, we always wanted to create a negative pressure environment because we wanted to know to sort of drive, instead of just creating just a physical barrier that would collect larger droplets of virus-containing spit and then allowing someone to then touch that or come into contact with that. We knew we wanted to always have sort of a negative pressure environment. And so with working with Clemson, we're able to now test these devices in a whole nother level. So what we've done is actually take the device prototypes and actually bring them into the clinical environment and test them in some of our negative pressure rooms at Greer Hospital, and actually underneath some simulated actual procedural conditions using particle testing with some of John's collaborators, Dr. Isham, I can't pronounce his last name, I'm sorry. Michella. And actually doing some of the particle testing, which is well above and beyond what has been published anywhere in the literature. Moreover, we're going to start publishing on some of the data of what actually goes on inside of a negative pressure room, which has been sort of just taken on faith that these work. And we're actually going to try to disprove some of that dogma, showing that actually the design of a negative pressure room in general may need to be rethought of in order to provide better protection for clinicians in the clinical environment. So I couldn't be happier working with the students. Amanda's on the call with us right here, who has worked tirelessly responding to texts from me in the middle of the night every time I have a new idea, and trying to revamp some of the prototypes and then get them in the environment and delivering them and using them. And we're using them clinically right now and generating data, getting some patient feedback and usability feedback from some of our clinicians and nurses right now in the emergency department with one of the prototypes. And so we're very excited about that. And as another side note, Amanda and her partner in the class as well, Robert Falconer, under the leadership of Dr. Desjardins and with some input from me, have also come up with some passive barriers that were being used by EMS and are still currently being used by EMS that have saved the overall health system countless thousands of dollars, in the sense that what was currently available on the market was a product as designed by Stryker, which, giving us a large discount, say of about 40%, was still over $300 a unit as a one-time use single barrier protection device that you would use and then throw away when transporting a patient through an ambulance from one hospital to another that was a positive COVID patient. The unit device that Amanda and Robert came up with is not even a tenth of that. And so the EMS division was very happy and very excited to work with Clemson on that limited project for something that was actually useful and then put into use. Subsequently, however, the FDA has revoked all the EUAs for just the passive devices. So we've had to try to back off where EMS is using those, only because they're no longer being allowed under an EUA license by the FDA. But it doesn't mean that they weren't responsive at the time to this crisis and putting something in place that at the time we thought was going to be a useful device, and did save the hospital thousands of dollars in using something. So.
Great. Well, thank you so much, John and Phil. That was excellent. And our final talk is with Dr. Larry Gluck, who will discuss a novel treatment modality that Dr. Gluck spearheaded. And again, the format, Larry, I know you came on late, is just to identify what the problem was, the solution, how the collaborations helped, and what inspired you to go about this. All yours, Larry.
Okay, thank you. Can you all hear me?
We can.
Okay, very good. All right. So there's a subset of patients infected with COVID who go on to respiratory failure, and indeed even multi-organ system failure, in which the drive may very well be a hyper-inflammatory response to the infection itself. So it's not so much that this is virologic destruction of the lung, but rather an unleashed, overreactive hyperimmune system that is creating respiratory failure. Right? So plasmapheresis, which is the use of a technique which we use multiple times every day to manage diseases such as Guillain-Barré, myasthenia gravis, or if we want to deplete abnormal cellular contents, acute leukemia, and the like. So it's a continuous loop from the patient where the blood is then subjected to centrifugal forces, and we can separate the blood into its component particles, its component parts, and then selectively deplete whatever is offending the patient. So if it's acute leukemia, we can deplete white cells. If it's sickle cell anemia, we can deplete red cells. And alike, if it's something which is contained within the plasma, we can deplete the plasma and replace with either FFP or albumin. So the thought process was that the respiratory failure may be coming from a wide spectrum of cytokine excess. And cytokines are large molecules, and the thought was, can we effectively deplete that with plasmapheresis or therapeutic plasma exchange? The world has been divided on this, where it's thought that patients with sepsis, for example, could potentially benefit from plasmapheresis, and those studies were largely inconclusive or negative. But we really had a thought that in this particular case, the cytokines that we had in mind may be capable of being subjected to plasmapheresis. So we have now completed 2 studies. The first study was a pilot study of 10 patients utilizing plasmapheresis alone. And what we found was that patients underwent plasma exchange 5 times over a course of about 8 days. And what we found was, we were able to substantially drop the cytokine load of multiple molecules of interest, as well as the marker CRP, or cross-reactive protein. Of interest, this directly paralleled the improvement in oxygenation as measured by a variety of indices, and these indeed paralleled each other. And in the first 10 patients, and again, we are selecting for the sickest of the sickest, with extraordinarily high mortality rates since the onset of COVID. And what we had was 7 out of 10 patients had major clinical benefit. All patients on high flow O2, up to 15 liters, as well as half of the patients on mechanical ventilation were able to be liberated from supplemental oxygen or ventilation in a matter of just a few days. The second study, which we have just completed, involved replication of the first 10 pilot, and then the use of a drug called Ruxolitinib, which is capable of inhibiting the JAK-STAT pathway, which is probably a major control point in the production of cytokine excess. So our theory there was, if we can acutely deplete the cytokines and couple it with a drug to turn off additional cytokine elaboration, that the combination may very well be better than just plasmapheresis alone. And we're analyzing that data, but we have just had a manuscript on the first 10 accepted for publication because of the success of this technique in restoring oxygenation. I think the important points were, we also were able to show that we did not compromise humoral immunity, that for those patients who had antibodies to COVID, plasmapheresis did not deplete them. The important points include the fact that this is a technology that exists widely throughout the world. And if you take a look at the onset of the pandemic, where some of the large cities were struggling, these machines were sitting in apheresis labs entirely untapped. And when we were looking for an intervention which could provide benefit to 10 or even 15% of the patients so effective, we were thunderstruck by how well and how many of our patients actually responded. For those who were on supplemental oxygen and could talk with us, and some of those could barely talk because they were nearing intubation, actually the patient reports were such that they said while they were on the machine, so they could actually sense a major improvement in their dyspnea. So we're talking about people who've been on the machine for 30 of what might be 60 minutes say, wow, I feel better already. So what we concluded, and this is an example of what goes in and what goes out, but really the highlights of what we have published is that we have shown that we can effectively reduce these cytokine loads. And this is extremely important because the cytokine loads also complicate other disease processes, such as the more common ARDS that takes incredible number of lives across the country every year. That we showed that indeed we can improve oxygenation, and that this is a technology which is ready, available, does not even need to be incrementally adapted for it to be effective. I think our opportunity is that because we have shown that we can drop the cytokine load and improve oxygenation in this disease setting, that this is going to translate into clinical trials for ARDS. It's going to translate into clinical trials for these things that complicate some of our immunological therapies, particularly CAR T. So we were happy to be able to generate this data so far, and we will be communicating the results of our second 20-patient trial in the not too distant future.
Great, Larry, that was fantastic. Thanks so much for that presentation. We have a few minutes for questions. I think there's one from John. I'm just going to read it and provide a quick answer, and others can pipe in and we can open it up. So how is Prisma Health doing health screening to frequently test large number of people healthy rather than treating people when they are sick, promoted by Harvard epidemiologist Mike Mira and others? So aware of that work. Very interesting, some modeling talking about frequent testing, quick response from answers in terms of not waiting 3 or 5 days for results, and accessibility. We do test asymptomatic people through some of our community events, and certainly our university partners are testing people frequently. We also know that that's a practice in different settings, whether in the government or in sport venues. We also know that that's not you still have to protect yourself, because even when you have a negative test, you still could be infected. And so certainly you still have to practice safeguards and so forth. We are embarking together with U of SC and Clemson and MUSC. USC is taking the lead on a statewide survey, which is going to be very exciting, so we can screen random households, thousands at a time, in 3 different time slots to see what the prevalence is of COVID over about a 9-month period. So that we'll hear some in November more widely. This project, we're looking for both asymptomatic infections through nasopharyngeal testing, as well as serology tests, including in children with finger stick tests. Let's open it up to questions. Michael, do you want to take that on? Or?
Why don't we do this, Dr. Littman? Why don't we kind of pull this, at least the formal part of this, to a close? So for those people who have 5 o'clock deadlines, but then for those who'd like, and as much as some of our participants can, we'll hang around afterwards and continue the Q&A, if that sounds good. So, excellent.
That's great.
Okay. So, first of all, Dr. Littman, thank you, and the group that you've assembled here. Thank you. Yes, if we could get a Zoom thumbs up across the board here, that would be great. I mean, your leadership on this multidisciplinary task force has been phenomenal. This has been very interesting, and the different things they've done. They're happening behind the scenes that Most of us
Yeah,
have no
and
idea.
honestly, Michael, it was the leaders of everyone who spoke. It's I really didn't have to do anything. It was just the creativity and leadership of everyone else. Great.
I'll give you that. Take whatever you can get, right? Well, thank everybody also for joining us today. This has been phenomenal. Amy, do you want to maybe give us a little preview on a couple things that are coming up, and then we'll at least close the formal session. And like I say, I encourage those of you who have the time and interest, stick around and we'll have some further Q&A. Amy?
Yes, I did send through chat a link to registration for our upcoming COVID-19 Meet the Finalist event. John Desjardins was, as he was talking, it occurred to me, oh, well, he'll be talking again in a couple of weeks about that cover. So he and Amanda, I think we'll see again, and you'll see others as well from Clemson. There are 3 categories within the COVID-19 because we had such a large response. So there's that event. We also have second of our last 2 Meet the Finalists on the 13th. That's small business category as well as education category.
Correct.
So we hope you'll mark your calendars and see us on those dates, 4 o'clock at Tuesday. And then the award celebration will be November 17th. And so the winners will be announced and all of the finalists will be featured on short, professionally made videos. This was fascinating. We could have done this for at least another hour from my standpoint. So thank you. It's been wonderful.
Absolutely. So thank you all. And let me turn it back to you, Dr. Litwin. Let's continue for those who are able to stay. Let's open it up to some further questions.
Great. Thank you, Michael and Amy. So any questions? I know it's 5 o'clock and some have other calls and may have other obligations, but feel free to just chime in or.
Dr. Litwin, this is Karen St. John. I'm not sure if you can hear me. Can you hear me okay?
Yeah, absolutely. Yep.
Okay. I had raised my hand earlier. When we had our conversation about the Rapid Innovation Task Force and how it had experienced the COVID outbreak and changed its way of operating so that you were much more responsive, in some ways changed your methods of collaboration. You had talked about that a little bit, and you had also you had used the analogy of Dunkirk. Is there any possibility you can recreate that and tell everyone else about that experience and how the analogy of Dunkirk fits? Because I thought that was just fascinating.
Sure. Yeah. Well, I mean, look, I certainly not a great historian, but I had seen the movie and knew a little bit about Dunkirk and was obviously very impressed at, incredible what happened. And I think one of the things that attracted me to Greenville, I've been here for 3 years, were the people and just the spirit of philanthropy and giving for no particular reason other than it seemed to be the right thing to do. And people, wanted to do that. And so that felt very comfortable. And then, coming into this crisis, in March 1st, it just, a lot of people just stepped up. And I got Jerry, incredible, friend, leader, just stepping up for no reason other than it's the right thing to do. You can't sleep at night if you're not doing the right thing. And there was really thousands of Jerrys, out there in Prisma Health and HSC. We could have featured 100 people, innovators who kind of reached out, and just, there's no grant, no, glory, no recognition, just the goal to help, to make a difference. Certainly it's personal for all of us. I've lost, 4 family and friends, many of us have. But, it's always personal, right? When you're in healthcare, and, our patients are like our family, and, we know that this is hundreds of thousands of Americans have died, and, we're on track to lose 100,000 more. And we feel like we've done a really good job, at Prisma Health, and our mortality rates are lower than a national average, and incredible the level of, everyone on this call really inspires me to kind of keep, just the level of creativity and genius, but just kindness, generosity, like Phil's talking about, waking up in the middle of the night and texting, that's not for anything other than, wow, I think I can solve this problem and make a difference. And Yeah, so I can open it up to others who might want to. Wes, I don't know if you have some thoughts about that, if you're still on.
I don't know if I could jump in and say, I found it really refreshing too that we had a lot of people that weren't being passive about this at all, both on the clinician side and then also on the Clemson side. It's not just like we'll just see what we can get as far as masks. We're like, no, we need to build our own mass decontamination program and test it and make sure it's safe. No, we want to make our own negative pressure rooms. We want to make our own ventilation split and device. It was definitely one of those things where I think we realized here in South Carolina that we might be on our own a little bit, in the same sense as that we wanted to make sure that we could take care of our own, and not rely on anyone else. And there's a certain frontier type of spirit, I think, that South Carolina has and wants to show that we can be innovators and stuff like that as well. And so I just want to highlight that as well, to say that we weren't going to sit by and be sort of passive or, hope that somebody will save us and swoop in and, demand that the federal government save us. No, we're going to take care of ourselves and try to make sure that we can come up with our own solutions in case that help doesn't come, which it might not.
Yeah.
One of the really amazing things for me as a clinician too is that our work with this task force, but also, largely within our institution throughout this pandemic, just kind of demonstrates that, while we're typically more siloed, it takes something like this to really break down those barriers. And, I've just developed tremendous now professional relationships with folks that I probably would not have interfaced with otherwise. And those types of linkages have everything to do with how successful we are with responding to future issues, to accelerating a lot of the projects and initiatives, and I think brings us together closer and more effective as just a community of well-intentioned folks. So I think, we've really kind of turned a bad situation into something very, very positive and helpful for our community and also for Prisma and our academic affiliation.
Yeah, and it wasn't just the Prisma Health frontline workers and the academic partners that were huge, but we had local business guys waiting outside, coming in with rolls of different products, saying, here, we're going to do this for nothing. Don't worry, we'll make how many you need, 30,000, 100,000, and Jerry was leading that, and Cody, but these guys would just show up with their rolls and their supplies and for the same reasons, and that's a big draw of South Carolina. I think it's a special place. Not to compare, but I came from New York, also a wonderful place, New York City, but they had a hard time. I mean, they got hit harder in early March, but there wasn't that same spirit of working together across hospitals, being able to bring patients from one place to another. We never had where we had to wear garbage bags. It wasn't even close, in fact, to where we have hundreds of thousands of masks in reserve in case, always planning for that the next level of crisis. Really, everyone's working really hard, and it's not just healthcare workers, but everyone who's part of the enterprise is there to help patients. So anyone have anything else to say about that?
Alan, I would just add really briefly that, in the early stages of this pandemic, there were so many things that were constantly in flux. And, when we looked at PPE, when we looked at CDC guidelines that came out when we experienced changes. And, the word rapid in the Rapid Innovation Task Force is it really truly was rapid. And that's something that I think is just, we can't minimize. We really need to just appreciate that we're able to very, very quickly accelerate just ideas into something tangible, and then translate that into something that could really help in terms of patient care and frontline protection. And even with just the basic eye shields that now we'll have a couple 100,000 of. Again, that came out of CDC guidance that just changed literally how we need to approach our PPE, and the fact that we could get something in that quantity so quickly is just astounding. So talking to my colleagues who are in Elmhurst, Queens, one of the worst hit areas, particularly in the emergency department, you know, we are just incredibly lucky to have the resources that we have.
Yeah, and Phil, I know you talked about the novel ideas, but Phil he led the Novel Ideas Committee, which really was allowed a place for everyone to kind of put forth. And we had nurses and providers from Toomey and from, you know, down in Richland, you know, giving different devices, and Phil and his team would vet it. And so it really was because we know that the solutions may come from the top or, you know, may come from the front line, but, you know, we want to make sure there's an avenue there where it's not just, you know, the leadership team kind of telling folks what to do, but it's really everyone's, you know, equal, and setting up those communication mechanisms, because obviously we need to have structure, otherwise it can get out of control. But I think everyone had a voice. Quick question around, I think Alexis Kelly had around who do you contact a small business. I would say Cody Reynolds would be the perfect person to contact for that. So do you want to give out contact information, Cody?
Oh, yeah, sure. My email, it's cody.reynolds@prismahealth.org. And I would just like to say that as Dr. Liao mentioned, the eye shields that we're currently getting in, that would not be possible without small businesses, because we cannot, or it's business in general, we cannot make those ourselves as a healthcare institution. We have to buy them as a third party, so we have to partner with someone. So Jerry Chang's business helped us make that possible, and it wouldn't be possible without him. And we're looking for other partners like him. I mean, we can't have enough partners that are interested in finding the next solution. So please let me know any opportunities that are out there. We're really interested in looking into them.
And Cody, perhaps you could put your contact information in the chat.
Happy to. One second.
Appreciate everyone's interest and certainly everyone who spoke. Really inspiring, and I think we need to keep telling the story, because it just the last part. And thank you, Amy, for kind of bringing forth that example, because that really is the spirit behind why we did all this work and want to continue to do this work in other areas and beyond COVID, as we hopefully someday soon emerge beyond COVID in the post-COVID world. Any final thoughts, Michael, before we close?
No, thank you again, Dr. Littman. This has been phenomenal. And I think part of the gap in questions was people are still absorbing. There was just so much. I feel like I was drinking from a fire hose here. So I think there needs to be some time for us just to absorb it and sort it out. But thank you all. Phenomenal. So we hope to see you again at a future InnoVision event. Thank you all for joining us today.